Cap Times Idea Fest 2020 is made possible by the generous support of our sponsors. Presenting sponsor, the Burrish Group at UBS, a financial services firm with global access and a local focus to pursue what matters most for its clients. Major sponsors are HealthX Ventures, backing entrepreneurs who are creating value with digital health solutions. Exact Sciences, pursuing earlier detections and life-changing answers in the fight against cancer. Quartz, health plans built with you in mind. And Madison Gas and Electric, your community energy company whose goal is net zero carbon electricity by 2050. Co-sponsors are Epic Systems and the Godfrey Kahn Law Firm. Other sponsors are Wisconsin Alumni Research Foundation, Home Savings Bank, Unity Point Health Meritor, Cargo Coffee, and the Forward Theater Company. Media partners are the Wisconsin State Journal and Madison.com, WKOW Channel 27, and Hinckley Productions. Please consider becoming a Cap Times member. Learn more at membership.captimes.com.
Hi, I'm Chris Murphy, Managing Editor of the Cap Times, and I'd like to welcome you to Cap Times Idea Fest 2020. This is the festival's fourth year, but the first time we're presenting it virtually. Our theme this year is 2020 Changes Everything, which is hard to dispute. With the COVID-19 pandemic, its effect on the health, education, and economic well-being of our community, as well as the calls for change from the Black Lives Matter movement, there's a lot to talk about. We think this year's lineup is our best yet. Idea Fest has grown into a signature event on the Madison calendar. It's also an important showcase for the Cap Times, our locally owned and century-old news publication. If you're not already, we hope you'll consider becoming a Cap Times member. As a member, you'll have access to special Idea Fest programs, plus benefits throughout the year. You'll also be supporting an independent and trustworthy local media source at a critical time. Find details online at membership.captimes.com. The session you're about to see about preparing for the next pandemic was put together by the Wisconsin Alumni Association, and we're grateful to them for doing so. We're also deeply thankful for our sponsors, including our presenting sponsor, the Burrish Group at UBS. So please, enjoy this session of Cap Times Idea Fest. You'll find the other videos too at captimesideafest.com.
Hello and welcome to the Cap Times Idea Fest. I'm Kelly Tyrrell and I'm a science writer and the Director of Research Communications at UW-Madison. This is not my first Cap Times Idea Fest, but it is my first virtual Cap Times Idea Fest, and I'm really thrilled to be taking part. I'm also here with Dr. Kristen Bernard. Dr. Bernard is a professor in the UW School of Veterinary Medicine, and she is an expert in virology and in emerging and zoonotic diseases, especially those carried by mosquitoes. She has expertise studying coronaviruses and having studied previous outbreaks of the SARS virus. Kristen, thank you so very much for agreeing to participate. COVID-19 has certainly presented a lot of challenges, but it's also presented a lot of opportunities.
Oh, thank you for inviting me. It's great to be here.
So I trained at UW-Madison as a molecular biologist, even though now I'm a science writer. And I first got to know you through a graduate school colleague who's now a virologist. And she spoke really highly of you when I first came back in my professional capacity. So I'm a fan, but maybe you could tell us a little bit more about yourself and what you study.
Yeah, I am a veterinarian as well as I have a PhD in virology. And I study primarily mosquito-borne viruses, and I try to understand how they cause disease. I use different animal models to do that. And for the long-term goal, it's really important to understand that basic disease to be able to develop vaccines and antivirals. So that's kind of the long-term goal.
Great. We're here, the name of our session is How Should We Prepare for the Next Pandemic? Because as probably most of us know, we're kind of in the throes of a coronavirus pandemic right now. Can you tell us a little bit about your previous work with coronaviruses in particular?
Yeah, when SARS, the very first SARS hit in the 2000s, I was at the New York State Department of Health at the time, and we very quickly ramped up to do testing, and that wasn't actually my role to do the testing at that point, but we started doing research to develop an animal model, and that is super important. You need that for developing vaccines, et cetera. So that was one of my main goals there. And I worked with another virologist who had been working on them for a long time.
I seem to recall back in January you were part of a panel. This was, I think, January 31st, so really sort of awareness of the significance of this pandemic, at least here in the United States. And something that you said at that panel really struck me, that 30% or so of colds are caused by coronaviruses, and most of us have had colds, sometimes several times a year, and they don't really, they maybe lay us out for a couple of days, but what is it about this particular virus, SARS-CoV-2, that makes it pandemic?
Yeah, so part of the way a pandemic happens is that it's new, and the whole population, we don't have any immunity to it at all, except for now the people that have been infected, of course. And so the cold coronaviruses, the ones that cause the common cold, there are lots of different viruses, by the way, that do that. But the ones that are coronaviruses, we've lived with those a very long time. So we have a lot of immunity in the population, in that so-called herd immunity. And so some people get sick every year from a cold-like virus. In addition, this SARS coronavirus that's circulating now is very easy to transmit between people. So it has a very high transmission rate. And then, as we maybe will talk about, people are shedding the virus and can transmit the virus even before they know they're sick. So it does have some unique characteristics there, although the cold coronaviruses also are very easy to transmit as well. But it's probably mostly because the whole population is not immune.
Right. So you said that you got started trying to make an animal model, basically a research tool to better understand SARS, which the initial outbreak, this was the SARS virus, right? The SARS coronavirus, not the same virus that we're dealing with now. And that was back in 2002, 2003?
Exactly right.
Did we have any inkling before that time that coronaviruses could become pandemic?
No, not really. I mean, at the time we were and we're always concerned about influenza pandemics because we've seen those historically, and we know that it could happen again, and we had the one in 2009, which was a mild one, fortunately. But we didn't really know that coronaviruses would be the next pandemic. We were nervous in 2002, 2003, but fortunately, it seems like that virus didn't transmit as well between people. I mean, I don't think we have really hard data on that yet. But we were fortunate in, and this is the global the whole world public health system was able to stop that virus. But we were nervous at the time.
Sure. Did anything, has anything changed since that time? I mean, have we been keeping an eye out for coronaviruses like SARS and the coronavirus that caused MERS in 2013?
Yeah, so definitely it's something that we've been thinking about because of that scare that we had in 2002, 2003. And then when MERS coronavirus hit, we were very nervous that it could become pandemic. And again, I think I keep harping on the fact that the ability to transmit between people is really what's going to drive a large pandemic. And fortunately, MERS, which in case people don't know, that stands for Middle Eastern Respiratory Syndrome, that one does not transmit well between people. It does, mostly close contact, people, healthcare workers, and close family members. So fortunately, we've never seen a big pandemic because of that, just small outbreaks. We did have a pretty big outbreak in Korea, and it does originate actually from camels. So camels transmit it to people, and then we get some transmission between people.
So it's really an animal disease that some people tend to get.
It is. It is.
I'd like to come back to that.
Okay.
But I think first I want to talk about what makes a good pandemic virus. And I know that's a horrible way of framing it, but you've talked about the transmissibility between people. So there are some characteristics of this current coronavirus that have allowed it to spread very quickly and make people sick around the world, which is what makes it a pandemic. Right. So what is it about a virus or another pathogen that would make it or render it pathogenic in a pandemic way? Yeah.
So again, high transmission between people, and I think also that ability to transmit before we know we're sick. That has been really a horrible outcome of this virus because it makes it difficult for, if you feel sick, you can stay home. Maybe your family members get sick, or you can even try to isolate yourself from your family members, but you can stay home. But if you are shedding that virus before you even know you're sick, that's a problem. And that's one reason why the masks and the social distancing is so, so important. And just to let people know, we are social distancing now.
Yes, we are.
In a very nice large room, so that makes us feel comfortable.
And we brought our masks.
And we have our masks. They're right here. Mine has branded with Wisconsin. Go Badgers. So yes, so that's very critical for a virus that it can transmit. And if we had, people have been talking a lot about that contact tracing, and the fact that it sheds for seems like several days, and also that there are people that don't even ever get sick and who are shedding the virus. Those two things make contact tracing very difficult, because contact tracing is going to depend on finding that person infected. Now, they could have just gone down and gotten tested, and even though they're asymptomatic, but not everybody's doing that. There's no reason to. Or if once they're sick, well, they've been shedding for how long, and they've been contacting how many people. So again, that's where, limiting your trips to the grocery store, not going to very large gatherings, things like that are really critical and make it very difficult to control this.
Right. So when you say shed, can you tell us what that means from a virology perspective?
Absolutely.
I think it's a word that gets thrown around a lot.
Yeah. So shedding, I talk a lot about that I teach veterinary virology, so I talk a lot about that. And right now I've been talking a lot about it to my students. And shedding would mean that you are actually secreting the virus. And for a respiratory virus, typically it's going to be in those coughs, sneezes. You can also get it into your mouth, because of course your airway and your mouth are connected in the back of your throat. So even if it's not shedding in your mouth, you actually have virus in your mouth, so saliva, and that's why some of those saliva tests are working. Coughing, sneezing, that kind of thing. And that's because the virus is actually replicating in the lining of your nose, your trachea, your lungs, and that then when it replicates, it produces the virus, and that goes into those secretions. So that's what we call by shedding.
Sure. So the virus is basically making many copies of itself in your nose or in your lungs, and then you sneeze and you emit it, and that's what helps pass it along to someone.
Exactly, and that's what we say is shedding.
Right. And that's one of the features of this current coronavirus, right, is that it is making copies in infected people, and they're going about the world because they don't know they're sick and they're carrying this virus.
Exactly right. Exactly right.
Okay. I think there's been some conversation about the SARS-CoV-2 virus and sort of where it came from. Mm-hmm. And we know that it emerged out of Wuhan, China at the end of or around the end of December. There's been some conversation about it potentially starting in bats. Can you talk a little bit about that and maybe bats as carriers of diseases?
Yeah. So these coronaviruses are really interesting. The very first SARS, as well as the SARS-CoV-2, that's short for SARS coronavirus. We say SARS-CoV because it's shorter.
And two because it's the
second one. And they're actually very closely related. That one from 2002, 2003, and the one we have now, and it's the pandemic virus, are very closely related. They both originated from bats. And the reason why we say that is because of other coronaviruses that we've isolated or been able to get from bats in the wild. They are genetically very similar, but they actually are not the same virus. So we know that the bats, it's not coming directly from the bats, and that there has been some other, what we think, another animal host, another animal that the virus has gotten into, has changed, circulated in that animal population probably for some time, has changed quite a bit. I mean, it's definitely not the same virus. And then that has gotten into people. And that we do link that still to the live animal markets in Wuhan. And that's probably where the original source came from.
Sure. So do bats get sick from coronaviruses? Are they, experiencing a pandemic of SARS-CoV-2?
Yeah, that's a great question. And no, we've not seen coronaviruses. Bats are pretty interesting animals. They have a different immune system, partly because of that hibernation that they go through. And we haven't seen any increase in mortality or anything like that in the bat population due to this. So we don't think so.
Sure. So basically the bats are, the word that I've heard is reservoirs for disease?
Yeah, a reservoir is a really good term for that. So when we say that, often reservoir means that they don't get disease. That's not always the case. We're sort of anthropomorphic, so we kind of think more, that animal species is that reservoir that's going to give us the virus, right? But often they're ones that do not get any disease at all. And that's common. There's lots of examples of that.
Sure. And so you talked about some other species, right, that maybe caught a coronavirus from a bat and then humans caught it from that other species. Mm-hmm. What is it about that species to species jump?
Yeah, so it's probably adapting. So it's possible that the bat virus doesn't infect people as well. And so it's probably adapting, and I don't know of any clear studies on this now, but definitely people are studying this kind of thing, that it's adapting in that other animal host that then allows it to replicate better in us. And we have that example also with influenza. Influenza virus is really a bird virus. So all the different influenza viruses that we have can infect birds. And only some of them infect mammals. And of the mammalian group, only some of them can infect humans. So what we think often happens, and when we get a pandemic, we get circulation in swine. So bird viruses, swine viruses, they recombine. It's actually called reassortment is the technical term for influenza. And then that gets into people. So there's that adaptation happening in another animal.
Sure. And I think we're talking about a disease that it's affecting a lot of people right now, but you're a veterinary researcher. You're in our veterinary school. And I think that might surprise people that a veterinary researcher would study human disease. But you've talked now for a few minutes here about the important role that animals play in disease transmission. And I mentioned a word earlier, zoonotic, that people might not be familiar with.
Yes.
So can you talk about what is zoonotic or zoonoses?
So zoonoses is any pathogen, so of course we're talking about viruses today, that can be transmitted between animals and people. And we in fact sometimes even call, if it goes from people to animals, we call that a reverse zoonoses, because again, us humans feel that we're more important, right? So any pathogen that can be transmitted from animals to people. Now there's some pathogens that stay in their species and we never see them go into another species or humans. But in this case of this coronavirus, of course, we've seen it go from animals to people. And then same case for some of the influenza viruses, and there's other examples. Prime example is rabies. Rabies virus is a zoonotic virus. It infects bats, dogs, cats, other mammals, and then can get into us.
Okay.
Yeah. But really, I would like to just add, you mentioned about how important animals are, and they really are. And that kind of brings up that concept of One Health, which I don't know if everybody's familiar with. One Health is that idea that both human health and animal health and the environmental health, are such key integrated players. And so then we come up with a concept that's One Health, that's health of everything. And it's critical to be thinking about all three of those things when you're thinking about trying to control diseases and make effect on public health.
Thanks, Kristen. I don't know that One Health is a term that I guess within the last couple of years became familiar with. I know because our veterinary school is really kind of forging the way in that arena. But it makes me think a lot about human interactions with wildlife. And maybe, you talked about the current coronavirus, the pandemic, being traced back to this live market in China. In this market, presumably there were wild animals in which people came into contact with, and it took one or a few people contracting the virus and then spreading it to other people. So would it make sense for humans to, shut down these markets and limit their interactions with wild animals that might transmit disease?
Yeah, I mean, that's a great question. And, I think interaction with wildlife and encroaching on their habitats is one way that we get emerging viruses. But I think we need to be careful when we talk about something like live animal markets in different countries. I mean, that's part of their culture. And, maybe there's steps that they could make that the Chinese government might decide, okay, well, we want to change the caging system or something different to try to minimize that contact. And I think that would be great. And I would love to see WHO working with China to do something like that. But I think we need to be careful talking about just shutting things down. It's a very strong cultural part of China and the Chinese people. I sometimes think, I've been thinking about this a lot lately, and I sometimes think it's maybe people in Wisconsin can appreciate that deer hunting is really part of our culture. And there are diseases that we can get from deer, such as chronic wasting disease, and things that we are doing that maybe are spreading that disease. And I don't think people of Wisconsin would want us to shut down deer hunting either. So there's things that we could do to try to control all these types of interactions with wildlife.
Sure. What is it about animals and the environment and interactions with people that maybe foster some of these transmissions? Or, maybe what I'm trying to say is we see a lot of virus and a lot of diseases sort of coming out of China, for instance, or parts of Africa. Ebola virus is something that people have heard of, and it's named after a river, right? What is it about these particular areas that are facilitating diseases in the human population?
Yeah, I think, and Tony Goldberg at the School of Veterinary Medicine studies this a lot. And so a lot of what I am going to be telling you are things that I talked to him about. And there's a lot more encroachment on the wildlife habitat these days. And so, especially in places like Africa and South America and things like that, I mean, it's not maybe a little bit different in China, where that encroachment means that we're having a lot more contact with these wild species that we never would have had before, or would have been a rare event potentially before. So now we're in contact with their pathogens, and that jump into a different species is more likely to happen. And I think that's the danger that we have, and part of that is changing the agriculture now that they're taking down forests and just getting up there. There's a lot of Tony Goldberg studies a lot that interaction right at the edge of the forest and the agriculture, and he sees a lot of interaction with non-human primates, and there is even more concern because of course they're more similar genetically to us, and there's a high chance of a jump.
Okay. Excuse me. I know one of the problems when you talk about these parts of the world where human needs and human economies are sort of butting up against wildlife and wildlife habitat. And one of the problems that you hear from some of these places is people get very fearful of wildlife and maybe start slaughtering animals in order to prevent disease. Can you talk about that as a strategy, or maybe why it's not a great idea?
Yeah, I mean, it's not a great idea at all, because we might, we're potentially decreasing the diversity of our animal population, and I think better. And then plus the slaughter, what are you going to do with those animals now? That's probably going to increase your contact with any pathogens they might have. And then just from an ethical standpoint, I think slaughter of animals just for trying to control something like that, I just don't think that's going to be an effective means either. So, there's ways that we can do by protecting their habitat rather than slaughtering their animals, protect their habitat and keep people from the interaction with animals. I think that's a much better way to go.
Sure. Is that part of this One Health approach that you've
Absolutely.
mentioned? Because you
Absolutely.
mentioned the environmental aspect of that.
Yeah, absolutely. So they're, having making sure that we keep good habitat, good diverse habitat is going to be essential in this One Health, absolutely. And then climate change also comes into that too.
Sure, maybe can you talk about that a little bit? What role does climate change play?
Yeah, so climate change, some of it is this change in the habitat. You know, we're getting a lot of decrease in frog populations and things like that. They're getting a fungal disease now, and some of that has been implicated due to the warmer weather. But there, you know, maybe we say, okay, well, it's just some frog species, maybe we don't care. But really, climate change is going to have a big impact on mosquito-borne viruses and tick-borne viruses. It has an immediate impact on how well the virus replicates in the mosquito. Higher, warmer temperatures allows the virus to replicate faster in a mosquito, such that that will cause more activity, more chance of people then getting infected by a mosquito-borne virus. Also can change the habitat of where the different mosquito species live. And for example, Zika virus and Dengue virus are transmitted by a tropical and subtropical mosquito species called Aedes aegypti. Now, if that, it is also another one they can transmit, it's called Aedes albopictus. Now, if those encroach into different regions, we could then have that virus in those other regions that we wouldn't normally have it.
Essentially, because it's getting warmer farther north,
then the mosquito can survive, and then also the warm temperatures are going to have higher replication rate. So with West Nile virus, for example, we have, that's in Wisconsin. So that's a mosquito species that is transmitted by a mosquito species that's actually in Wisconsin. Throughout the United States, actually, these mosquitoes, different mosquito species. But in that case, we already have the mosquito species in this region, but that warmer temperature will mean that we have a higher virus activity in that particular year. And we've seen that. We see spikes of West Nile virus, and it's mostly due to weather. Some of it's due to some other factors as well, but weather is a big factor there.
Yeah, what is West Nile virus?
West Nile virus. So it's related, so we always, as a virologist, we always have to talk about the families. So it's a flavivirus, which is related to yellow fever, dengue virus, Zika virus. Those are probably ones that everybody's heard of. And it is a mosquito-borne virus that emerged in 1999 in New York City and then spread across the United States. By 2004, actually, it spread so fast, and as a virologist, we were very surprised at how fast it spread. It spread through the 48 contiguous states and then into Central and South America, and a little bit encroached into Canada as well. So that virus is spread between mosquitoes and birds, so it cycles and is in all these different regions that I just talked about, in birds and mosquitoes in every year. But we only see disease, or we really only talk about the disease if it's in people or horses. They both are affected quite severely.
Would West Nile classify as a pandemic virus or no?
We called it an epidemic at the time because it was not worldwide. But it was definitely an epidemic in the United States. Now we actually call it endemic. And those terms mean, endemic means we now have it here. And it's at a certain level. We might see in some years, as I talked about, little outbreaks. Maybe we call that a little epidemic in Wisconsin or an epidemic in Texas, wherever it happened to be. And then a pandemic would be worldwide. But we don't see West Nile virus worldwide. There are some restrictions on where it is.
Sure. One virus that you mentioned, or maybe two, Zika and dengue, those are both, they're present throughout the world, right? With climate change and with change of range of mosquitoes, might we see those diseases here in Wisconsin at some point?
I think in Wisconsin it would be unlikely to get a lot of Zika or dengue, but in maybe even as far up as Illinois or something like that, because this is a the mosquitoes, we do see occasionally Aedes albopictus in Wisconsin, but we don't really see it over winter. So both of these mosquitoes don't do well in our winters. Now, 40 years from now, could that happen? Maybe. You know, I think it depends on really how bad our climate change gets. I think in the next 20 years, it would be unlikely to see it in Wisconsin. It all is going to depend on that mosquito vector. And those mosquito vectors are actually really interesting because those are both dengue and Zika. They are cycling between the mosquito and humans. So I talked about West Nile cycling between mosquitoes and birds. These Zika and dengue actually is between mosquitoes and humans.
So when you say cycling, you mean it's passing from one to the next?
Yeah.
And what is the virus doing?
Yeah. So, well, I love mosquito-borne viruses, I guess you can tell. So it's really interesting. So the mosquito is going to pick up the virus when it feeds on an infected bird in the case of West Nile or humans in the case of dengue and Zika. So let's just stick with dengue and Zika because it's a little easier. So when they feed on that infected person, if that person has the virus in its blood at the time, the mosquito gets it in its blood meal, and these are always the female mosquitoes, by the way, and that virus will then replicate in the mosquito, replicates in the gut, so in the intestines of the mosquito. It then spreads into the mosquito body, gets into the salivary gland of the mosquito, where it replicates, and then is transmitted the next time that female mosquito needs to lay eggs. She'll feed on blood again because she needs all that protein for her eggs, and she'll pick up the the virus in the blood. No, I got ahead of myself. She'll then feed on a naive person and deposit the virus at that time. And some of my studies is actually to understand if the mosquito saliva has an effect on the disease process, and actually it does. So I find that very fascinating. But these viruses are so interesting that they replicate in an invertebrate, an insect, and a vertebrate, a bird or a mammal. And so they have to be very adaptable.
And that's because they have different kinds of cells, right? And viruses need to basically recognize something on the outside of a cell in order to get inside, basically to connect to it and sneak in.
Yeah, absolutely. And so it's really remarkable that these viruses evolved to be able to do this. And they're also replicating at different temperatures. If you think about the mosquito is at room temperature, right, or ambient temperature. And, mammals are at 37 degrees Celsius, so typically 37 to 39.
Viruses are pretty talented.
Yeah, unfortunately, right?
Yeah, and sneaky.
Yeah, they're very much so.
Okay, well, we're going to need to take a quick break, but when we come back, I'd love to talk more about how we look for pathogens that might become pandemic.
Sounds good.
Cap Times Idea Fest 2020 is made possible by the generous support of our sponsors. Presenting sponsor, the Burrish Group at UBS, a financial services firm with global access and a local focus to pursue what matters most for its clients. Major sponsors are HealthX Ventures, backing entrepreneurs who are creating value with digital health solutions. Exact Sciences, pursuing earlier detections and life-changing answers in the fight against cancer. Quartz, health plans built with you in mind. And Madison Gas and Electric, your community energy company whose goal is net zero carbon electricity by 2050. Co-sponsors are Epic Systems and the Godfrey Kahn Law Firm. Other sponsors are Wisconsin Alumni Research Foundation, Home Savings Bank, Unity Point Health Meritor, Cargo Coffee, and the Forward Theater Company. Media partners are the Wisconsin State Journal and Madison.com, WKOW Channel 27, and Hinckley Productions. Please consider becoming a Cap Times member. Learn more at membership.captimes.com.
Thanks and welcome back. Kristen, you mentioned your colleague Tony Goldberg, and he's working at sort of this wildlife-human interface. And we talked a little bit about people looking for different coronaviruses and influenza viruses. Can you talk a little bit about how we, and I'm using this as sort of the ubiquitous we, but how are we keeping an eye out for or surveilling for viruses that have pandemic potential? And then maybe can you talk about what pandemic potential means?
Yeah, so let's start with what pandemic potential means, I guess. So to me, pandemic potential is going to mean a virus that has a potential to infect humans and that we don't have immunity to it. All right, that's really going to be where that pandemic part of it's going to come from. So the human population in general has very little or no exposure to that virus before. That will potentially get a pandemic. And of course then be able to infect a human and transmit between humans. So those are I guess the three things you would need to have a pandemic virus. And we definitely are doing a lot of surveillance as the global we. But Dr. Yoshi Kuoka, also at the veterinary school, you'd mentioned that there's a lot of us at the veterinary school that work on these viruses. He is involved in a lot of studies where they're doing surveillance in swine, in people, in birds. As I mentioned, the influenza can replicate in birds and swine and other species as well. But those are the ones that we're most worried about with the potential to get into humans. And they are doing a lot of studies where they're, first of all, sequencing the viruses that are collected, and they have a lot of collaborators across the world really doing this work. And we know so much about the viruses now that there are some particular mutations that worry us more that would potentially allow a virus to go human to human. And some of that work has been done by doing animal studies and looking to see what allows the virus to transmit between animal models, such as a ferret, for example. And if we know what those are, we then know what mutations we should worry about if they come up when we do this kinds of surveillance where we're looking for all the viruses throughout the world in different animal species. So that's one area, and that's kind of very directed towards influenza virus. The other viruses, it's hard to know what's going to be the next pandemic. Of course, we worry about things that we already know has that potential now, SARS. Still worried about MERS. Maybe it could change such that it would have higher human-to-human transmission. And Ebola, some things like that. So we're looking to see, are we starting to get an outbreak in a human population? Can we then stop it? So we're watching for that disease now in humans. But the next virus that we don't have any idea about it being a pandemic makes it a little more difficult. These days, a lot of people, and some of the work that Tony Goldberg does, and others around the world, we're just sort of surveilling the wildlife populations for different viruses. And just even knowing a lot about those viruses is helpful because we then, if they did become pandemic, we already have kind of a base research knowledge about it. And that, if we would even be further behind if we didn't already know quite a bit about SARS and MERS coronavirus, the first SARS coronavirus, with this pandemic. So having that basic research has really helped.
Sure. I imagine that prior to the SARS outbreak in 2003, there probably weren't a lot of people studying coronaviruses. Mm-hmm. Did that change? And then have we kept up that sort of, if there was a higher level of scientific inquiry to try and better understand that virus, did we keep it up since 2003?
Yeah, so that's a really great question, and it's always interesting as a scientist to kind of watch the interest in a particular pathogen, especially when you work on something like a virus. And so we always had some people working on coronaviruses. We didn't have that many. I'm thinking back before 2002, 2003, but we always had some. Some of them were just a mouse hepatitis virus is an example of one. And probably nobody's heard of that. But that was some very basic work that was being done and by a handful of people. There's also a cat virus called Feline Infectious Peritonitis virus, and some people were studying that. So we had some basic knowledge about coronaviruses. But there wasn't a ton of research being done. When SARS coronavirus, the first one, happened, there was an explosion. This is a higher level containment virus, and So it did limit some of the researchers that were able to do it, but there was a lot, NIH put in a lot of money to fund different research.
And by higher containment, you just mean that it's more risky to study in a lab setting.
Yeah, so there are different levels of containment for, depending on the risk to the researchers or to the general population. And so SARS coronavirus, the first one, is at BSL-3 level. And actually, infectious work with this current pandemic one is also at the same level. But then after a few years, people were doing some basic research on vaccines and animal models, as I mentioned, the kind of work that I was doing back in the early 2000s. And then a new thing comes along, and less people are doing the research, and it gets harder to fund if the virus is no longer around. But there was always some basic research. I have some very good friends who continued to work on SARS coronavirus and then MERS when it happened. And they were always saying, this could happen again, this could happen again. But the funding dropped way down. But we did always keep some. And we would have been so much further behind if they hadn't kept some work going.
So I think what I'm hearing is investing in basic science is maybe a little important.
Yeah, investing in basic science is essential, especially because we can't predict when these pandemics are going to happen. And so just having that knowledge base is so important. We get so far ahead. I like to tell my students when I'm teaching about the example of Dr. Howard Temin, and maybe some of you have been on the lake path.
Howard Temin Lakeshore Trail.
Exactly, and I like to run down there a couple times a week, so I see a little plaque to him. So Dr. Temin and Dr. David Baltimore, they discovered reverse transcriptase, which is the enzyme that HIV and other retroviruses have. And they discovered that way back in the late 60s. And we didn't even know about HIV until 1980s, I think around 1980. My dates are not always the best. But if he hadn't known already about retroviruses and this particular way that they replicate, we'd been so far behind when HIV hit. So that just seems to me it'd be a really important example of how basic research that none of us knew would be important in a human disease like HIV.
What role is that investment or that baseline in previous coronaviruses playing now? It strikes me that this virus, SARS-CoV-2, was completely unknown to us, right? Just 9-10 months ago, I guess almost a year ago if we consider back in December in China. But we're watching scientific discovery happen in real time at sort of rapid pace, and it does seem like information changes every day, and what we know about the virus changes every day. So, what can you say about the sort of the pace of science and what we should take from what we're hearing on a daily basis?
Yeah, I think that makes it really difficult as the general public because information is changing so rapidly. I think we did know quite a bit about coronaviruses, just to back it up a little bit. And I'd like to say it's amazing to think that we knew about the sequence, and within like 2 months, NIH and Moderna, had this potential vaccine that they were starting to put out. So, I think that's all amazing, but that is how fast that part's happening, even just the development of the vaccines. And our understanding, we didn't know anything, and this is not reacting like the first SARS. Back in the panel that you talked about in January and in early February, I was making assumptions it was going to act like that. And so I was so hopeful, and I did say I was hopeful, I think back then, that we wouldn't have a pandemic.
And by act like that, that's what you mean, not become pandemic, not be as easily transmissible between people. We could contain it.
We could contain it, exactly. That we would be able to contain it and use the same types of measures, public health measures, to contain it. And clearly that didn't happen this time. But those kinds of things and how it's transmitted, that there are people that are asymptomatic, no disease, that are shedding the virus, as we talked about earlier. All those things we're learning in real time. And so our recommendations early on maybe we don't need to wear masks, but then we find out, wow, this is shedding with people prior to being sick and also who are not sick at all, that the masks then are helpful. The social distancing is helpful. And so all those things we're learning, that information that we're giving to the public is changing some because we're learning more. And I think for people in the public, to understand that we are, and that's the way science does work, that we learn something, somebody else learns something else and builds on that, and there's just a constant building and exchange between scientists and the scientific literature. And it can be very frustrating, I think, as a public member, but I think that's the way science does work, and I hope maybe the public can be a little patient with us.
Sure, so it's not that scientists are wrong per se, right? It's that science is a process and it's iterative.
Right.
It seems like we are in a moment where we need to make a lot of policy decisions based on science and the science that's evolving quickly. What can we do, especially since this session is about preparing for the next pandemic, how can we really foster that interaction or that dialogue between the scientists in a rapidly changing environment and the people who make decisions about what we do?
Yeah, I mean, I think that is a very difficult thing, and I'm hoping that we're learning from this pandemic. I mean, I'll just say that I never thought we'd see a pandemic like this in my lifetime. So I think we are learning a lot about that interaction. I think having a really strong message from people like Dr. Tony Fauci, director and also director of CDC, you know, those people that are really in touch with the current science, that is really critical, that we then make policy from that evidence rather than let the policy dictate what the evidence is or dictate what we should do. It really should be evidence-based. And, you know, things are changing, and people like Tony Fauci are extremely smart, and he can realize that things are changing and we need to change our policy. But we need to have a clear message.
Sure. And this is probably not the last pandemic we'll see, right?
No, I don't think so. I hope not. I mean, I hope that we don't see another one, but I'm sure we'll see more outbreaks and epidemics of things, but we'll hopefully never again this bad.
Yes. Yeah, you mentioned influenza. I know that the influenza virus it's different than the coronavirus. Maybe we can talk a little bit about how they're different and what makes influenza a virus that we are concerned about on a yearly basis, right? I mean, I know that the World Health Organization has a surveillance program. The CDC is involved in those efforts. You mentioned Professor Kawoka and some of the research that he and some of his colleagues are doing. So what is it about the flu virus that requires that we remain vigilant about that?
Yeah, and I also like to shout out Wisconsin State Lab of Hygiene. They're also a center of this flu surveillance and, of course, all the testing that's been going on with coronavirus. So they give the sequences to CDC. So all of our Wisconsin influenza sequences are going to CDC through them. And then Dr. Koch is doing more of the animal surveillance. So flu is really different. And the reason why we need a vaccine every year is that it actually will change and mutate its outer proteins. And you had kind of talked earlier about how a virus interacts with a cell in our bodies or another animal, and it adds a specific interaction. And that specific interaction, our antibodies that we make can actually disrupt that. But if the flu then changes a little bit, so no longer do the antibodies bind to it, it can now infect the cells again.
And we get these antibodies from having the virus and recovering from it, right?
Absolutely.
Or from taking a vaccine that encourages our immune system to make, the antibodies are essentially defenders, right?
Right, absolutely. So that's one of our defenders in our immune response or our immune system are those antibodies. And they're really important in protecting us from an infection. There are other parts of our immune system that are really important for clearing the virus, but antibodies are really important for, sometimes they keep us from getting infected at all. So they're really very powerful. And yes, so that virus, we will have antibodies because we got sick with flu last year or potentially from the vaccine that we took last year. But then as it's moving through the population, the virus, the influenza virus, it's changing its outer surface. And it's got pressure to do that because I'm anthropomorphizing the virus now, it wants to be able to transmit between people. It has to survive. So it's changing that outer surface such that when it gets into the next person, their antibodies don't react against it. And flu is just, again, anthropomorphizing, an expert at that. That's its modus operandi, is to mutate enough, and it does that every time, slowly accumulates these mutations such that the vaccine is no longer effective next year. And so the surveillance is critical, and that's what CDC and WHO are doing, is to see, oh, it's changed like this, so now we want our vaccine to have this particular virus so that we can be protected from that next time. And they're looking at the southern hemisphere, because they're having their winter, they're ending their winter now, and we're starting our winter. They're looking at those flu strains there, and then making an assumption that we'll be having those flu strains in the northern hemisphere in our winter. So there's a lot of assumptions that are going in, so sometimes the vaccine isn't perfect because we have to make it before we really know which viruses are going to be circulating in the northern hemisphere, as if we've talked about North America. So they, sometimes we'll say, oh, the vaccine's not so great this year, and that's just because there's not a good match for what's circulating.
And it's not a good match to the specifics of the specific identity of that virus and the tools that it has to essentially bind onto a human cell and cause an infection.
Exactly right.
Sure.
Exactly.
Now you've mentioned the word sequencing or sequence a couple of times. Mm-hmm. Can you maybe talk about what it means to sequence a virus?
Yeah, so sequencing a virus is to actually determine what that genetic code is. So all viruses have a genome, just like we do, all of our chromosomes, but their genomes are much smaller. The coronavirus is a fairly large one. That one's about 30,000 nucleotides. I mean, that's hardly anything.
And the nucleotides are like letters of the alphabet,
letters of the alphabet, that A, C, G, T that you see that makes up DNA. Those are those letters. And the code works exactly the same as it does for us. We have that code, that genetic code that we'll then be able to make proteins from. So that's exactly the same because they're actually using our cells to do that. So we can sequence that, just like you've probably heard of sequencing, people sending off their DNA to get their whole genome sequenced. And so we do that for viruses as well. And that helps us trace where a virus has originated, as well as those mutations that I talked about. If we know what mutations are important to potentially transmit the virus better or escape the immune system, escape those antibodies, that gives us a lot of information. And we can do that so much more quickly now. When I was a graduate student, it used to take me days to get about a couple thousand nucleotides, and now they do it just like that. So.
That's amazing.
Yeah, it is amazing. It's just amazing. I feel like I like to tell my graduate students, well, I used to have to pour these gels and it took me so long. It's just like walking uphill both ways without any shoes in the snow, It's like, oh gosh.
And now you're sitting on the beach with...
Yeah, now I'm just looking at my computer screen, here, give me my bases.
That's really fascinating. It's interesting how the role that technology plays in our ability to monitor for, surveil for these viruses. And I've worked quite a bit with Professor Kawuka, who you've mentioned, because one of the things he does is he is looking at the sequences of of influenza viruses that are circulating in avian populations, so in birds, usually waterfowl and other wild birds, or in swine and pigs. And he's looking for those particular strings of letters that might make the virus more likely to spread between people, right? And you mentioned ferrets as an animal model because they somewhat mimic our modes of transferring viruses, right?
Right.
And so So essentially, one method of surveilling is to just keep an eye out for this sequence in the population, right? And what, and I imagine this is the case for other kinds of viruses. You're looking for particular sequences that exist in nature. What does that mean for us? Like, let's say we start looking for these sequences, or we are, and we find it. What does that mean from a preparation standpoint? You mentioned vaccines, but what about, medications or further investment in particular types of research? How does sort of that technology and our ability to sequence and look for these things affect what we can do from a preparedness standpoint?
Yeah, some of it is understand. So fortunately with influenza, we know so much about those various genetic code, those mutations, and whether or not those are important in transmission. But for a lot of our other viruses, we don't know that. We can track that we have certain mutations, but we don't know, we say, the phenotype. We don't know what is going to what the result is going to be of that. Will it allow it to be more transmissible? And for example, with the SARS coronavirus 2, we really need to know those things, because if it became even more transmissible, that would be something we need to, one, maybe change the vaccine, but two, try to stamp out that particular outbreak. Let's not let that particular virus go widespread. So I think that's one thing, especially with influenza and any other times that we know a virus that has that kind of capability to be more transmissible, that we could then concentrate our efforts and say, okay, in the case of a human coronavirus, public health message needs to be that you need to stay at home. We need a stay-at-home measure here. We need to be wearing masks at all times. Let's reduce people going into work, et cetera. So that's the kind of information that I think, knowing not just the sequence, we need to also know what that is the result of that sequence, what the phenotype is.
Essentially what it does, what it
right.
allows
what
the virus
does.
to do.
Exactly,
yeah.
exactly.
And probably, you told me a very interesting story about a couple of viruses in a region in Africa affecting palm fruit, or involving palm fruit. And I guess I'm thinking about the fact that, we've talked about some fairly drastic things, like developing a vaccine, which, as we've seen on a pretty profoundly, is challenging. It's happening remarkably fast.
Remarkably fast.
Taking dramatic actions like, closing down parts of our economy and shutting down schools. But I think what struck me about what you told me about these couple of other viruses is not all actions to sort of contain or stop spread need to be super dramatic. Can you tell me that story?
Yeah,
again?
so this is a really interesting story. It's actually in South Asia.
Oh, I'm sorry.
Yeah, no, it's okay. So there are two related viruses called Hendra and Nipah. And Hendra emerged in Australia in the 1990s. We just see occasional disease with Hendra virus. It's in fruit bats, and it will infect people and horses, and people and horses have disease. But the Nipah virus emerged in Indonesia, actually. And we saw it first in swine. So here's that idea that, doing surveillance is so important. Here we had a large respiratory disease outbreak in swine, as well as death. And then simultaneously, people were starting to get sick. Anybody that were swine caretakers, as well as abattoir workers, were getting sick and getting actually a brain disease. And so the public health system and WHO figured out that it was a brand new virus, and that virus is called Nipah. And Nipah virus, then we figured out, was closely related to Hendra. Again, all this sequencing helps us. Well, then the things that we knew about Hendra helped us learn more about Nipah. Because they were related, we said, oh, is there a bat source? And sure enough, it was in fruit bats. And then we started seeing, so that really was controlled in Indonesia fairly well. But we started seeing this disease in people in South Asia. And it turns out that it was connected with people drinking unpasteurized palm juices and eating some of the palm fruit. And what happens is these are fruit bats. So fruit bats like to eat fruit. And the palm juice, they would actually put a spike into the palms and collect the juice, just like we do for maple syrup. All right, and it would be high up in the tree. People climb up and put that spike in there. And then they have a clay pot that collects that sap. And the fruit bats would come in, especially at night, and drink straight from that. I mean, why not? You know, that was easy fruit to get for these fruit bats. And they would urinate in the collection vessel while they were eating. And it turns out, we talk about shedding virus, well, this virus is shed in urine of these fruit bats. So then people were getting sick by drinking this unpasteurized palm juice. And that's the way they liked to drink it. They didn't want to pasteurize it, so that would be one way to control this. But instead, what they did is they climbed up and put bamboo shields around those containers so that the bats couldn't urinate it in anymore. I mean, and it just was such a simple solution to this problem that I really loved. You know, that I think public health worked with the farmers or collectors of this palm juice, and you know, what can we do? You know, and that sort of also shows that, you know, we don't always have to have that really high level technology to fix something, and it's really important to talk to the actual people that are being affected.
Absolutely, absolutely. We've talked a lot about people, but we haven't talked as much about diseases that are pandemic in animals, and maybe we don't use the word pandemic, but we rely on animals for a lot of things. We're in Wisconsin.
Yeah.
The dairy industry is obviously a very important part of our economy. So especially as a veterinary researcher, what kinds of things are we concerned about in animal populations?
Yeah, that's great. And actually it's called panzootic, just in case you wanted to know, or epizootic. Yeah, there's definitely a lot of viruses that we're concerned about in our veterinary species. And if you have a dog or a cat, you've taken it to be vaccinated, and some very serious diseases are controlled by those vaccines, including rabies, of course. But we have an example of another coronavirus that I could tell you about that caused an epidemic about 6 years ago in the swine population. And we'd never seen this virus before. It's called, now I'm forgetting, Porcine Epidemic Diarrhea Virus, PEDV. And
I can't believe you forgot that.
Yeah, I know. So it actually emerged in Iowa and Indiana in the swine population. We've never seen it before. Caused a lot of death in the swine farms. It turns out it actually was a virus that was from China. So it had been described in China swine populations before, and it's now basically in the U.S. swine population. We do have a vaccine for it, though, so fortunately we can control it. But those kinds of things happen quite frequently. There's another virus that there's always viruses that we're concerned about getting into our livestock population that we don't have in the U.S. or in North America. A good example is foot and mouth disease virus. We're always worried about that. So if you've gone through, and the USDA, APHIS asks you questions when you come home from being abroad, have you been on a farm? That's the main reason, is that foot and mouth disease virus. But then when there's other outbreaks happening, and in fact there's one now called African swine fever virus that's in China, causing a huge number of deaths in the swine population there. And that is a big concern for the USDA and the swine farmers here in the US. So there'll even be more questions if you were at those areas where there's activity for those viruses. And it really affects trade. It's tremendous impact on the economy.
Yeah, well, I am very, very grateful to people like you and our Wisconsin Veterinary Diagnostic Lab looking out for the animal
shout out
diseases.
for them too.
But yes, I'm very grateful to the scientists, to you, to your colleagues who are keeping an eye out and trying to keep us safe and helping respond to our current pandemic and get us closer to a vaccine or a way of going about life maybe slightly differently for the foreseeable future, or maybe forever, but helping us get there a little bit sooner. Kristen, thank you so very much for being here.
You're very welcome. Very welcome. Enjoyed it.
Yes, thank you. And thanks to all of you for joining us. Enjoy the rest of your Cap Times Idea Fest.
Cap Times Idea Fest 2020 is made possible by the generous support of our sponsors. Presenting sponsor, the Burrish Group at UBS, a financial services firm with global access and a local focus to pursue what matters most for its clients. Major sponsors are HealthX Ventures, backing entrepreneurs who are creating value with digital health solutions. Exact Sciences, pursuing earlier detections and life-changing answers in the fight against cancer. Quartz, health plans built with you in mind. And Madison Gas and Electric, your community energy company whose goal is net zero carbon electricity by 2050. Co-sponsors are Epic Systems and the Godfrey Kahn Law Firm. Other sponsors are Wisconsin Alumni Research Foundation, Home Savings Bank, Unity Point Health Meritor, Cargo Coffee, and the Forward Theater Company. Media partners are the Wisconsin State Journal and Madison.com, WKOW Channel 27, and Hinckley Productions. Please consider becoming a Cap Times member. Learn more at membership.captimes.com.